Oh no, not this again. Yeah, it's this again. The problem with studying anything in just the abstract, like we did here, is that it remains so abstract, it just kind of loses some meaning. I'll give you an example. What do you mean by DNA? Well, DNA. It's the chromosomes. Yeah, well, what DNA? Oh, it's the genes. Sure, but what genes? There's 20,000 genes, and they're in every single cell. So which one do you mean? I don't know, just pick one. Well, here's the point. It's not just one gene, and it's not all of them. Each cell expresses probably somewhere between 10 and 30% of the genes within it. 10 to 30% of that 20,000. That's not a lot, but it makes sense, right? That's how a brain cell and a liver cell become different, because they express different parts of the genome. That makes sense. The questions that automatically arise from this then is, well, how do we turn the other genes off? And also maybe, how do we turn on a gene when we need it, and then turn it off when we don't? Can we do those things? Well, sure. Let's take a look. All right, well, do you remember what these arrows stood for? This one was what? Transcription, and then translation. Right, you got it. So, where can we control gene expression? Transcription or translation? Well, the answer is yes. Either one. Probably makes more sense if we're going to stop gene expression to stop it here. Otherwise, it's a real waste to make RNA if we're never going to make the protein. If you're a bacterium, that's pretty much the only way you control genes, is you just don't make the RNA. You don't do transcription. We do a little bit of both, but a lot of it is here. So let's take a look at different ways we can stop, or at least control, transcription.
Probably the simplest way of controlling gene expression is to just coil up the DNA to make it physically inaccessible to RNA polymerase. So when we've talked about molecular genetics, RNA polymerase comes in and acts on the DNA. When we talked about things like mitosis, we looked at the whole chromosome. Here it's duplicated into sister chromatids getting ready for division, and that helps it to fit inside the nucleus and be moved around without getting tangled up. But RNA polymerase can't get into this really coiled up DNA. That just doesn't work. And so if you coil up the DNA too much, you cannot express those genes. On a daily basis, we can actually change this, believe it or not. We can coil and uncoil DNA. It might look like this. So this little puffy segment here where you see the strands of DNA, that is where it has been uncoiled, taken off the histones, and now RNA polymerase can get in there and transcribe those genes. The rest of this stuff is still super coiled and is kind of off limits to RNA polymerase. So just that physical act of coiling is one way of controlling gene expression. Let me show you a different and kind of unusual example of this, where that becomes a little more permanent. This only applies to females, in this case, because females have 2 X chromosomes. Now, they don't always operate with both of those X chromosomes. Very often, one will be supercoiled and remain supercoiled in something called a bar body. You can actually see that over here in this diagram on the right, where one of them is supercoiled up down here. Okay, so how does that relate to this cat, and what does that have to do with calico coloration? Let's think about it this way. Somewhere along the line in development, you had this cell, and it was far enough along that says, okay, we're big enough, we need to inactivate one of our X chromosomes. We don't want to use both. So that cell divides, and that decision is made, and the mom's X chromosome stays active here, and the dad cat's X chromosome remains active there. And that decision remains more or less permanent, so any of the cells that divide again over here all are expressing the mom's X chromosome, and the ones produced by that cell express dad's. In the calico cat, that results in these patchy fur colorations. Maybe the dark patches are from dad's X chromosome and the orange patches are from the mom's. And this is why the calico cats are only female, because only females have two X chromosomes. Males can't be calico. But the larger point is that by coiling up DNA and keeping RNA polymerase from getting in there, we can change the expression of those genes. Well, DNA coiling is all well and good, but that doesn't answer the question, how does a cell know? Now, if you're just a single-celled organism, then you just produce one cell and that's you. But for multicellular organisms, where we start out as these stem cells, cells that have the potential to be all different kinds of cells. And along the way during our development, those stem cells differentiate into bone cells and fat cells and nerve cells and so on. How do they know how to do that? Well, they need to coordinate with one another. And part of that is done genetically through these master genes, where one gene makes a protein that goes out and controls the other genes, which then signal out other genes and says, look guys, here's how we're going to work together. I'm going to express this set of genes, and so my cells are going to do this. And so we can see in this developing fruit fly, which we'll come back to in a moment, these different colored segments where different master genes are being expressed. So a lot of this work was done at Columbia University in the early 1900s by Thomas Morgan in his lab, and that's why you often see fruit flies associated with this. They would develop mutants. They would irradiate these fruit flies, knock out certain genes, and then they'd get weird things like antennas growing where the legs should be, or legs instead of eyes. Some really weird stuff, and through that figured out that the body plan of these fruit flies was determined by some specific master genes called Hox genes. And so these genes that control other genes basically say, here's where our tongue should be if that gene is produced. And over here, this is going to be the abdominal segment corresponding to the green area here, saying that's where we're going to express the genes that make our guts and this body segment. And so master genes are genes that control the expression of other genes. Again, getting at transcription. Express this gene, not this gene. And that's how we develop into a complex multicellular organism. The last way that I'll talk about that affects transcription is epigenetics, overwriting our genes. And so this does not change the DNA sequence, but it does effectively shut off genes. And so for a variety of usually environmental reasons that are listed here, our cells will add usually a methyl group, just a chemical group that is added to the DNA. So it's known as DNA methylation. When you hear DNA methylation, that's one form of epigenetics, and a pretty common one. It can add that chemical group either directly to the DNA or also onto the histones, the proteins that DNA gets wound around. And usually what that does is it turns the gene off. This can have a variety of bad outcomes, from cancer to autoimmune diseases, in us if we just go around and start semi-randomly shutting off different genes. The point is, this affects transcription. We have the right allele, but we no longer use it. RNA polymerase passes over and says that gene is not accessible right now. And so that's another way that gene expression can be affected, and it's related to human health. And this is a pretty new field, one that we're just starting to understand well.
Well, there's a few ways we can just outright stop transcription or kind of turn it on and off. There's another one in that sort of weird in-between stage in eukaryotes like us. Remember when we got RNA just fresh off the press and we had those little bits, those segments that were introns and exons? We had to cut out the introns and then ditch them. Well, that actually provides us the opportunity to do some weird stuff. It's called alternative splicing. So that cutting out was splicing, And normally we put this exon and this exon together and we'd call it good. But what if we did this one and then this one and then this one? We could come up with different combinations and that would make different proteins. And we do that actually in making antibodies, for example. So that's kind of another interesting semi-related note. Let's move on to translation and talk about a couple of ways we might control gene expression there. And we'd probably do this if we know that we want to make a protein within a cell, but we don't want to wait. So we want to have the RNA ready to go and not wait as long to make the protein. We don't want to start from scratch, in other words. Let's take a look at those.
First of these has to do with a new kind of RNA called microRNA. We're familiar with mRNA, and we can see that being transcribed from the green DNA here. That's a familiar process. But now we have this new microRNA. Maybe that is transcribed from a different gene in the DNA somewhere. When you look at that, you can tell that the microRNA and the mRNA have complementary bases there and there. When you think about what would happen in that case if these got to a big ribosome, well, this starts to get fed through, but now the microRNA creates kind of a speed bump in a way, and it cannot be translated. So this is an interesting way that cells have of delaying or even stopping the transcription or the translation of proteins in the cell. Well, let's look at yet another one having to do with ribosomes, and there's a few here. So translation, remember, takes our messenger RNA and makes these proteins. So some pretty obvious ones. At some point, we don't keep this mRNA around forever, and so the cell is going to degrade this, and the faster the cell does that, then the fewer proteins than we have around. In the same fashion, if we start chopping up these proteins once we're done with them, that in a way effectively stops that gene expression because it stops the protein from being used. The title here about ribosomes being inhibited by tetracycline is a bit of an aside, but related to this, saying now that we understand the process of gene expression well, and we know what ribosomes do, we can understand one type of antibiotic. Now, antibiotics work in a variety of ways, and tetracycline, its unique sort of mechanism is that it stops translation at the ribosome. But it does it in a really specific way. Bacteria have different types of ribosomes than we do. And so tetracycline affects bacterial protein synthesis, but it does not affect the protein synthesis in our own cells. And this is what makes a lot of antibiotics so great, is that they affect bacteria, but not us. And so this takes advantage of that difference in ribosomes in order to inhibit the protein synthesis and therefore life process of bacteria while leaving our cells mostly alone. Now, side note here, since our mitochondria are similar to bacteria, in fact, they have the same types of ribosomes, with enough tetracycline, they can be affected, and this can start to make us tired and really wear down, so we have to be careful about the dosage and length of time that we take some of these. Kind of an interesting quirk of evolution there.
Well, there's translation. We can even go one step beyond that and say, I've made my final protein. I have the amino acid sequence, it's been folded up, and now I have a whole protein made and ready to go, but it's actually still kind of off, right? I haven't let it go yet. We talked about this with enzyme regulation. So this is basically coming back around to the same thing, saying, I have a protein, and another way I can activate that is, for example, to add a phosphate group, which changes this active site, and now that protein is activated. So we can even wait till the very last minute, and that still kind of falls under, at least in a broad sense, this idea of gene regulation. Well, there's gene regulation. It's taken this abstract idea, this central dogma that DNA codes for RNA, which codes for proteins, and said, let's be more specific about that. Which of those 20,000 genes am I going to express in a cell? and that's going to determine the character of my cell. Is it a skin cell or a brain cell? Is it a liver cell or something lining the large intestine? All of those are going to require different genes, and our body has to have some way of controlling that. Bacteria even do this themselves. You'll learn more about their particular style when you get to microbiology later on. It's an important process, but kind of impressive that a bunch of molecules in a nucleus can somehow sort out which genes need to be expressed and which ones don't. Life is pretty amazing, isn't it?